What's going on, besties? This is everything you are going to need to know all in one place for the ATI TEAS version 7 portion of the exam, specifically the life and physical sciences. Let's get started. So when we think about biological systems, they're structured across multiple layers. Each one is integral to understanding the entirety of life processes. The first level that we're going to explore is the cell. It's the most elementary unit of life. But is there anything smaller than a cell? Well, yes, indeed there is. Within cells that house organelles, these organelles are smaller than the cells themselves. Moreover, we have biomolecules, which play crucial roles within the cell and are tinier than the cells themselves. And even further down is we have atoms. We can break those down further into subatomic particles, which are again even smaller than the cells. However, for the TEAS, we're going to begin at the cellular level, which is the fundamental unit of life. This is the universal truth, regardless if you're a bacterium, a fungus, protist, plant, or animal, all living things are going to be cellular in nature. When these numerous cells collaborate to perform unified functions, they form what is known as tissues, marking the subsequent level of biological organization. Cardiac muscle tissue, for example, consists of cardiac muscle cells working in concert together to make our heart beat. These tissues can then combine to compose organs, representing that third tier of our hierarchy of biological structures. Your body is composed of numerous organs, such as your heart, your lungs, and your kidneys. When these organs collaborate inside of our body, they are going to form what we call an organ system. Let's take the digestive system for example. It encompasses various organs, including our stomach, our small intestine, large intestine, and liver. They all work together in harmony to aid the digestion of food and the conversion of that food into energy necessary for cellular processes. The digestive system is merely one instance of an organ system. When you interconnect all of these organ systems together, what emerges? An individual, an organism, just like you. There are additional biological hierarchies, including population, community, ecosystem, biomes, and biospheres. But for the purposes of the ATI TEAS, this is the group that you should focus on. Our first practice question is, in the context of biological organization, which of the following sequence correctly orders the hierarchy from simplest to the most complex? Is it A, B, C, or D? And the correct answer is A, cells, tissues, organ systems, and organisms. As we know, the correct answer is always going to start with our cells. That's the basic unit of life. Cells group together to form tissues, which combine to create organs. These organs work together in organ systems. And then, of course, multiple organ systems are going to create the organism. That is what we are. Our next question states, which statement accurately describes the relationship between organ systems and organisms in biological hierarchy? Is it A, organ systems exist independently of organisms and do not interact with each other? Is it B, an organism is composed of multiple cells, bypassing the need for an organ system? Is it C, organ systems are composed of organs that work independently to support an organism's life? Or is it D, organisms are comprised of multiple organ systems working together to ensure survival? And the correct answer is D, organisms are comprised of multiple organ systems working together to ensure survival. If these systems did not work together, we wouldn't really survive very long. So yes, the correct answer is going to be D. Now that we have a basic understanding of biological hierarchy, let's get down and dirty when it comes to cells. Modern cell theory encompasses several key principles. Firstly, it states that cells represent the most fundamental unit of life across all organisms. Secondly, it asserts that every living entity is composed of cells, which may be singular, like we see with bacteria, which is a unicellular organism, or it could be made up of multiple cells, making them multicellular like humans. The third principle states that cells arise only from existing cells, highlighting their capability to replicate. When you think about it, cells are complex entities that house genetic material and are capable of division. They have internal structures or organelles that perform various functions and processes. On Earth, cells are categorized by two principal types, reflecting the diversity of life's cellular foundations. Within the cellular world, you are classified as either prokaryotes or eukaryotes. Bacteria and archaea all both fall within the prokaryote category, while fungi, protists, animals, and plants all fall within the eukaryote category. Both of these categories share common features, such as genetic materials, cytoplasm, and ribosomes. Each are small organelles responsible for protein synthesis. Additionally, they both have cell membranes that regulate the passage of substances in and out of the cell. However, the distinction between them is pretty significant. Prokaryotes suggest that with the prefix pro, meaning no, that's going to be a memory trick for you to remember for the ATI TEAS, is that they lack a nucleus that houses and manages genetic information. Furthermore, they do not have membrane-bound organelles, which are more complex structures, like a nucleus, mitochondria, and Golgi apparatus like we find with our eukaryote cells. So whenever I'm trying to remember which one has a nucleus and which one has membrane-bound organelles, I use this mnemonic. Pro means no, which means they don't have those, and U means do, which means that they do have those. You might now be curious about the roles of organelles. What functions do they serve? Well, let's begin by talking about the cell membrane, also referred to as the plasma membrane. This barrier has selective permeability, which means that it allows only specific materials in and out of the cell. The selective entry and exit point helps maintain stability within the cell and state known as homeostasis. Once we get inside the cell, we are immersed with a jelly-like substance known as the cytoplasm. This substance envelops all of the cell's internal structures, and it's present in both prokaryotes and eukaryotes. Contrary to what one might assume, the organelles floating within the cytoplasm receive substantial support. This support comes from the cytoskeleton. It's an intricate network of fibers that not only provide structural support for the cell, but it also plays a crucial role in cellular movement. Navigating our way through the cytoplasm, we encounter ribosomes first. Notably, ribosomes are not enclosed by membranes and are found in both prokaryote and eukaryote cells. Their primary function is protein synthesis. This is a critical process since a significant portion of genetic material, also known as DNA, is dedicated to coding for proteins. Ribosomes may either float freely within the cytoplasm, or it can be anchored to another organelle, which is a topic we're going to discuss very soon. Shifting our attention to organelles exclusive to eukaryotes, we now explore those that are encased in membranes. This ultimately brings us to our key player, the nucleus, often considered the control center of eukaryotic cells. It's here within the nucleus that our DNA is housed and stored. While all cells do possess DNA, in eukaryotes it's neatly contained inside the nucleus. The nucleus oversees the cell's operations and houses the nucleus, which is a specialized area where ribosomes are assembled. Next up, the endoplasmic reticulum adjoining to the nucleus's membrane or nuclear envelope plays a crucial role in the cell's operation. This organelle is going to be instrumental in processing molecules such as the folding of proteins and serves as the primary conduit when it comes to transporting throughout the cell, much like a cellular highway. The endoplasmic reticulum comes in two forms. We have rough ER, which is studded with ribosomes, lending to the name rough in its appearance, and we have smooth ER, which is going to lack those ribosomes. The rough ER is particularly active in the synthesis and transport of proteins. It's a direct consequence of its association with ribosomes, which are the cellular machinery responsible for protein synthesis. Molecules exiting this ER is often enclosed in vesicles that bud from the ER itself. On the other hand, our smooth ER has a much broader range of functions, including detoxification, which is a vital process especially abundant in liver cells due to their high content of having smooth ER. Additionally to detoxification, the smooth ER is involved in synthesis of certain types of lipids, further diversifying its functional portfolio. Moving on to the Golgi apparatus, often regarded as the cell's premier packaging and distribution hub. It accepts materials from transport vesicles that have detached from the ER, employing enzymes to modify these receiving molecules and organizing them accordingly. The Golgi apparatus also decides the destination of these molecules, including directing some of the cell membrane for secretion, effectively expelling them from the cell. Amidst all of these cellular activities that are taking place, you might begin to question what exactly is fueling them. Enter the mighty mitochondria, which is a single mitochondrion if we're referring to just one. These organelles function like cellular power plants, generating ATP energy through a process known as cellular respiration. Unlike conventional power plants, mitochondria operates on glucose. It's a type of sugar and requires oxygen to produce ATP energy efficiently. In addition to mitochondria, plant cells boast remarkable organelles known as chloroplasts. Chloroplasts are capable of synthesizing glucose through the process of photosynthesis, which harnesses light energy. Their green appearance is attributed to the pigment that they absorb from light energy, reflecting green light back, giving plants their colorful characteristic. Lysosomes, often dubbed the cell's waste disposal system, are membrane-bound organelles filled with enzymes that break down biomolecules, such as proteins, lipids, and carbohydrates. They play a crucial role when it comes to digesting cellular debris, unwanted materials, as well as invasive organisms like bacteria, maintaining cellular cleanliness and recycling materials that are reused by the cell. Vacuoles, on the other hand, are large, fluid-filled sacs found predominantly in plant cells, though they are also present in some animal and fungal cells. Their functions are going to vary, including storing nutrients and waste products to maintaining internal hydrostatic pressure within the cell and sequestering harmful materials. In plants, vacuoles are pivotal when it comes to growth because they absorb water and expand, enabling the cell to grow larger with minimal investment of new biomass. Now let's do some practice on everything we just learned. Which of the following cell components is primarily responsible for assembling proteins using instructions encoded in mRNA? Is it the nucleus, ribosomes, Golgi apparatus, or mitochondria? And the correct answer is B, our ribosomes. So again, our ribosomes are those cellular structures responsible for protein synthesis. So they actually read the sequence of mRNA and assemble amino acids into proteins based on what was coded in the instructions. The nucleus houses that DNA, the Golgi apparatus modifies and packages the proteins, and the mitochondria are involved in energy production. But ultimately, it's those ribosomes that are going to be directly related to synthesizing proteins. Which organelle is known as the powerhouse of the cell due to its role in producing ATP, which is the cell's main energy currency? Is it the chloroplasts, the nucleus, mitochondria, or endoplasmic reticulum? And the correct answer is C, mitochondria. Like we talked about before, that mitochondria is responsible for producing ATP, also known as adenosine triphosphate. They do this through a process of cellular respiration, which is why they're often referred to as the powerhouse of the cell. Our chloroplasts are involved in photosynthesis when it comes to plant cells. Our nucleus is involved in ribosome production inside our nucleus. And of course, our endoplasmic reticulum is involved in protein and lipid synthesis as well as processing. So let's talk about the differences between mitosis and meiosis. Initially, I approached learning about mitosis and meiosis as two distinct topics. A direct comparison laid out side by side would have been significantly better and aided me in understanding it, which is why I'm going to teach it like this today. So what are the key differences? Well, mitosis is actually going to lead to the formation of somatic cells, also known as body cells, while meiosis is going to produce reproductive cells known as gametes, which is sperm and egg cells. Let's consider the initial state of a cell involved in both processes. Both are going to start off as a diploid cell designated by 2N. This just simply means that they contain two complete sets of chromosomes. For humans, this means that one set of 23 chromosomes were inherited from their mother, and the other set of 23 chromosomes was inherited from their father, totaling a total of 46 chromosomes. During the interphase stage, the cell replicates its chromosomes. Even after duplicating 46 chromosomes, the chromosome count is still referred to as 46 because the copies, known as chromatids, remain joined at the region called the centromere, effectively doubling the chromatid number to 92. And although interphase does precede both mitosis and meiosis, it's not technically part of their process. However, it is a crucial phase for chromosome duplication, setting the stage for the ultimate cell division that we're going to discuss. And by the way, illustrating a full 46 human chromosomes can be a little challenging. So for the sake of clarity and ease visualization of our diagrams, we're only going to use 6 chromosomes to help us understand these concepts. So in order to help us understand cell division, we're going to use the acronym PMAT, which stands for the sequences of both mitosis and meiosis. However, the only difference is that meiosis includes these stages twice, resulting in each phase being designated with a numerical suffix, like we see with prophase 1, metaphase 1, so on and so forth. While we're going to outline the basic events that occur during the PMAT stages for the ATI TEAS, it's really important for you to understand that everything we're talking about is barely scratching the surface of all the intricate details that are taking place. When it comes to the ATI TEAS, this is what you're going to need to know. So during mitosis, prophase is going to be that initial stage when it comes to division. It's indicated by the prefix of pro, which means before. This is when chromosomes become more visible as they condense, ultimately becoming thicker. In contrast, prophase 1 of meiosis features not only chromosome condension, but they're also going to start pairing up in homologous chromosomes. These homologous chromosomes are roughly equal in size, gene type, and location. One came from the mother and one came from the father. This stage is going to allow for the exchange of genetic material between chromosomes through a process called crossing over. This is going to lead to the creation of recombinant chromosomes, which are essential for genetic diversity. Next up, we have metaphase. So in mitosis, that nuclear envelope that was previously enclosed in the nucleus has already been dismantled by the time that this phase begins. A mnemonic when it comes to metaphase is remember that M stands for middle, as during this phase, the chromosomes are going to align in the cell center, forming a single row. In contrast, in metaphase 1 of meiosis, while chromosomes are gathering in the cell's central area, they're going to remain in those pair of their homologous counterparts. Therefore, the alignment is not going to be a single row, but rather a pair of chromosomes standing together in the middle. During anaphase of mitosis, a helpful mnemonic to remember that is to associate A with away. At this phase, the chromatids are going to be separated and drawn to opposite ends of the cell by spindle fibers. In the context of meiosis, anaphase 1 is going to mirror this concept, but with a crucial distinction. It is the chromosomes rather than the chromatids that are going to be moved away to opposite ends of the cell. In both mitosis and meiosis, specifically telophase 1 of meiosis, chromosomes are going to reach the opposite sides of the cell, and the process of forming new nuclear envelopes around these chromosome sets are going to begin, setting the stage for the creation of 2 brand new cells. Each new nuclei is going to encapsulate the chromosomes on each side, paving the way for the cell to eventually divide into 2 separate entities. Following that nuclear division stage is cytokinesis. That is the partitioning of the cytoplasm is going to finalize that cell division process. Thus, at the conclusion of mitosis and cytokinesis, 2 genetically identical diploid cells are going to be produced, both having 46 chromosomes in humans. This process is crucial for an organism's growth, as generating more cells is going to be essential for development and for the repair and replacement of damaged cells. Well, that's it for mitosis. Let's move on to meiosis part 2. So in prophase 2, we observe that the chromosomes are condensing within both cells. This phase is less eventful compared to prophase 1, primarily because there's no homologous pairs anymore undergoing crossing over in this stage. During metaphase 2, the mnemonic M for the middle applies once again. However, in this phase, the chromosomes are going to align in a single row down the middle, similar to the arrangement that we saw in metaphase of mitosis. In anaphase 2, the mnemonic A for away is going to happen, and in this stage, the chromatids are going to separate themselves and are going to be drawn to opposite sides of the cells. Remember, during the first stage, they were still chromosomes. Now they are separating their chromosomes into chromatids. So this is huge when it comes to meiosis anaphase part 2. During telophase 2, chromosomes are going to be moved to the furthest opposite ends of the cell, and the formation of new nuclear envelopes are going to begin to encapsulate these new divisions, leading to the creation of new cells. Following the completion of meiosis 2, cytokinesis is going to occur, dividing the cytoplasm entirely. This is going to mark the conclusion of meiosis, resulting in 4 genetically distinct cells, or gametes. In this process, males are going to produce sperm and females are going to produce egg cells, all of them being haploid cells carrying half the chromosome count of the original cell. For humans, each of these gametes contains 23 chromosomes. Notably, the fusion of sperm and an egg cell creates a diploid cell, or a fertilized egg known as a zygote. This zygote is going to undergo numerous mitotic divisions, leading to the development of a new organism. Let's do some practice questions. During which phase of meiosis does crossing over occur, contributing to genetic diversity by exchanging genetic material between homologous chromosomes? Is it A, prophase 1, B, metaphase 2, C, anaphase 1, or D, telophase 2? And the correct answer is A, prophase 1. That crossing over occurs during prophase 1 of meiosis. This is when those homologous chromosomes pair up and exchange segments of genetic material, leading to genetic variation in resulting gametes. Which of the following statements accurately distinguishes between mitosis and meiosis in terms of genetic outcomes? Is it A, mitosis results in 2 genetically identical diploid daughter cells, while meiosis produces 4 genetically unique haploid cells? Is it B, both mitosis and meiosis result in 4 genetically identical daughter cells? Is it C, mitosis produces 4 genetically unique diploid daughter cells, whereas meiosis results in 2 genetically identical haploid cells? Or is it D, both mitosis and meiosis produces 2 genetically unique diploid daughter cells? And the correct answer is A, mitosis results in 2 genetically identical diploid daughter cells, while meiosis produces 4 genetically unique haploid cells. We talked about this a little bit before, that mitosis, when it comes to cellular division, is going to result in those 2 genetically identical diploid daughter cells essential for growth and repair. And with meiosis, on the other hand, that is a 2-step division process that's going to result in those 4 genetically diverse haploid cells, also known as gametes, which is going to be crucial for sexual reproduction. In meiosis, homologous chromosomes segregate during anaphase 1. What is the significance of this event in terms of genetic variation? Is it A, it ensures that each gamete inherits an exact copy of each chromosome, reducing genetic diversity? Is it B, it results in the duplication of chromosomes, increasing the chromosome number of gametes? C, it allows for the random assortment of chromosomes, contributing to genetic diversity among offspring? Or is it D, it eliminates the need for crossing over as a means of generating genetic variation? And the correct answer is C, it allows for the random assortment of chromosomes, contributing to genetic diversity among offspring. So during anaphase 1 of meiosis, homologous chromosomes are pulled apart to each end of the cell. This segregation allows for that random assortment of chromosomes, meaning that each gamete is going to receive a mix of maternal and paternal chromosomes. This is really significant when it comes to assortment, because this random assortment is going to contribute to that genetic diversity among the offspring, as it produces a variety of potential genetic combinations. So let's talk about heredity. So heredity explores the transmission of traits from parents to their offspring, a complex process that cannot be fully appreciated without a fundamental grasp of DNA, chromosomes, genes, and traits. Let's consider each one of us whose physical characteristics such as body patterns and size are determined by the genetic information coded in our DNA. In addition to DNA, environmental factors can also influence inherited traits. For example, our growth could have been impacted with insufficient nutrition. Contrary to the idea that DNA is a singular hidden code, it's actually a widespread code present in the nuclei of almost all of our cells, inherited from both our mother and our father. Another important note is that some animals have the ability to reproduce asexually, meaning that an animal could have received all of its genetic material from just one parent if that were the case for some species. Nevertheless, the essence remains the same. DNA is responsible for coding the traits that define us. It's essential for cell function, and it influences everything we've come to know, such as our height, eye color, risk for certain diseases, and hair color. Let's talk about the structure of DNA. It's not only beautiful, but it's also pivotal in understanding the mechanism of inheritance. DNA, which also stands for deoxyribonucleic acid, falls under the category of nucleic acids, one of our essential biomolecules. Composed of building blocks known as nucleotides, nucleic acids feature three critical components: a sugar, named deoxyribose, a phosphate group, collectively forming what is often referred to as the sugar-phosphate backbone of DNA, and most importantly, we have a base. It is the sequence of these bases that encode the genetic information that we've come to know, directly correlating to the traits an organism exhibits. In DNA, we have four types of bases, commonly abbreviated as A, T, C, and G. A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. These bases pair up in a specific manner, and a well-known mnemonic can assist in remembering these pairings. We have apples in the tree stands for adenine is going to pair with thymine, and then cars in the garage means that cytosine is going to pair with guanine. This base pairing rule of these DNA bases is universal, applying to all living entities, from animals to plants, protists, as well as humans. However, the total number of DNA bases, as well as the sequence in which these bases are arranged, is going to vary depending on the different species and even individuals within those species, underpinning the diversity of life. DNA consists of two strands, with nucleotides aligned along each side of them. In the center, the bases of opposite strands are going to pair up, connected by hydrogen bonds. The structure of DNA is going to twist into what we know as a double helix shape. Segments of DNA constitutes genes, which means specific regions of DNA corresponds to individual genes capable of encoding proteins. We call these structural genes. These proteins are going to play a crucial role when it comes to trait expression. Take human eye color, for example. It's a complex trait influenced by multiple genes, which direct the production of proteins responsible for the pigmentation that we have in our eyes. However, proteins are going to influence far beyond just eye color. It's also going to encompass transport, structural support, enzymatic activity, which is facilitating the synthesis of various substances, defense mechanisms, and so much more. It's also important to note that not every gene is going to be involved in protein synthesis. DNA also contains non-coding regions. These are what we like to call regulatory genes. Despite each of our body's cells containing a complete DNA code, only specific gene segments may be utilized, with certain genes being activated and deactivated through various mechanisms. This process is known as gene regulation. So your body contains a vast amount of DNA, which when compacted is organized into structures we know as chromosomes. This organization is particularly useful during cell division, as it simplifies the process of distributing DNA into new cells. Chromosomes consist of DNA coiled around a protein scaffold. Humans possess 46 chromosomes, which means that almost every cell within our body carries the same number of chromosomes. However, when we talk about human gametes, which is our sperm and our egg cells, these particular cells only carry 23 chromosomes each. Consequently, you inherit 23 chromosomes from your mother and another 23 chromosomes from your father, which gives you a complete set of 46 chromosomes. This constitutes your unique genetic code. Let's do some practice. Which of the following best explains the relationship between DNA, genes, and chromosomes within the context of heredity? Is it A, genes are made up of chromosomes, which are specific segments of DNA that code for proteins? B, chromosomes are composed of DNA and genes are specific segments of chromosomes that dictate individual traits? Is it C, DNA replicates to form genes, which then organize into chromosomes to be expressed as traits? Or is it D, chromosomes replicate to produce genes, which are then transcribed into DNA to determine hereditary information? And the correct answer is B, chromosomes are composed of DNA and genes are specific segments of chromosomes that dictate individual traits. So remember, chromosomes are very long strands of DNA wrapped around proteins called histones. Genes, which are segments of DNA, are located on these chromosomes and they're going to act as instructions to produce proteins. These proteins are going to determine the characteristics or traits that can be passed from parents to offspring through heredity. Which of the following best describes the function of regulatory genes in gene expression? Is it A, they encode for proteins that are directly involved in metabolic reactions? Is it B, they provide the code for structural components of the cell membrane? Is it C, they produce proteins that help in the replication of DNA? Or is it D, they produce proteins or RNA that controls the expression of other genes? And the correct answer is D, they produce proteins or RNAs that control the expression of other genes. So regulatory genes are responsible for producing proteins and RNAs, and they play a crucial role when it comes to controlling the expression of those genes within an organism. So these regulatory molecules can either enhance or inhibit the transcription of target genes, thereby regulating gene expression in response to the cell's needs or external stimuli. DNA often seems to hog the spotlight, with its stunning double helix structure resembling that spiral laddered appearance. Indeed, DNA is crucial for storing genetic information and determining traits, yet the significance of RNA frequently goes underappreciated. RNA is essential for conveying genetic instructions to cells for protein production, a process explored in depth when discussing protein synthesis. Ultimately, it's a biomolecule just as pivotal as DNA, and according to RNA world hypothesis, RNA might have even predated DNA. When comparing DNA and RNA, it's notable that both are found across all forms of life. In eukaryotic cells, DNA is typically located inside of the nucleus, whereas RNA is going to be present inside and outside of the nucleus. Unlike DNA, which has a double-stranded appearance, RNA is typically single-stranded, presenting with only one strand of nucleotides. The sugar component differs between the two as well. DNA contains deoxyribose, which aligns with its name, deoxyribonucleic acid, indicating the absence of an oxygen molecule. That's where the deoxy at the front of deoxyribonucleic acid comes from. In contrast, RNA's sugar is ribose, reflecting in its name, ribonucleic acid. When it comes to nitrogenous bases, RNA is going to include adenine, uracil, cytosine, and guanine. Did you notice something different? Uracil replaces thymine found in DNA. This substitution is going to call for a little bit of a tweak from the familiar mnemonic that we learned about base pairings. So instead of apples in the tree, we're going to say apple under the tree. That way it lets us know that adenine is going to pair with uracil when it comes to RNA. When it comes to car in the garage, that's still going to remain the same because we still have cytosine and guanine pairs that are going to pair together when it comes to RNA. It's crucial to understand that while DNA does hold the instructions for your traits, it cannot be expressed without RNA's intervention. RNA plays a key role when it comes to translating DNA's genetic code into proteins that carry out numerous functions within our bodies. There are 3 different types of RNA that's going to be important for you to know because of their roles. mRNA, also known as messenger RNA, conveys genetic instructions derived from DNA. In eukaryotic cells, while DNA predominantly resides in the nucleus, mRNA possesses the unique capability of exiting the nucleus. This allows it to transport the genetic blueprint to the ribosome, the cellular site for protein synthesis. Notably, RNA is also a significant structural component of ribosomes themselves. RNA found in ribosomes is known as rRNA, short for ribosomal RNA. Another crucial type of RNA is tRNA, which stands for transfer RNA, whose primary function is to ferry amino acids to the ribosome, ensuring that they align correctly with the corresponding mRNA codons. As these amino acids are sequentially linked, they form a polypeptide chain. Proteins, which play a myriad of roles within an organism, are composed of one or more polypeptide chains. Protein synthesis unfolds through 2 principal phases: transcription and translation. A useful mnemonic to recall this sequence and steps involves noting the letters C in transcription and L in translation. Since C precedes L in the alphabet, it's easy to remember that transcription is going to occur before translation. So starting with transcription, it's going to involve converting DNA into a messenger strand. This process is going to take place within the nucleus where the DNA is going to reside. During transcription, the enzyme RNA polymerase attaches matching RNA bases to the DNA template. These RNA bases are then linked together to create a single-stranded molecule of mRNA. Messenger RNA is composed of an RNA sequence that mirrors the DNA template. It's important to note that mRNA typically undergoes substantial editing before it's ready for action. This editing process is both intriguing and essential when it comes to the correct functioning of the protein synthesis mechanism. As we had previously noted, one of the most notable advantages of being mRNA, especially in eukaryotic cells, is the ability to exit the nucleus. mRNA is going to travel from the nucleus into that cytoplasm where they're going to associate themselves with ribosomes. Ribosomes, which are responsible for protein synthesis, are comprised of rRNA, which the R, remember, stands for ribosomal RNA, making that mnemonic pretty straightforward. The next phase we encounter is translation, and this is where the ribosome is going to assemble the protein. So let's simplify the essentials of what occurs in the cytoplasm. There you're going to find numerous tRNA molecules at the ready. tRNA, which stands for transfer RNA, is responsible for carrying amino acids, which are the fundamental building blocks of proteins. As we're in the process of synthesizing proteins, these amino acids are crucial for construction. The assembly of a protein requires the aggregation of these amino acids, which is a task orchestrated by our tRNA. But exactly how does tRNA know which amino acid to collect? This is where the role of mRNA becomes crucial. The mRNA is going to serve as a guide determining which tRNA needs to be brought to the ribosome and consequently which amino acids need to be assembled to form our protein. So each tRNA molecule is going to search for bases that match its own mRNA strand, aiming to find a set that is going to be complementary. Upon locating these matching bases of the mRNA, the tRNA is going to contribute its amino acid to the growing protein chain. This matching process is going to involve reading that mRNA sequence in groups of 3 bases at a time, not individually. These groups of 3 are known as codons. For instance, tRNA molecules are going to encounter a codon of AUC of the mRNA, while having a corresponding anticodon of UAG. Any tRNA with its anticodon is designated to carry the amino acid isoleucine. When a tRNA possesses that UAG anticodon pairs with a UAC codon on the mRNA, it donates that isoleucine to the polypeptide chain. After the transfer, that tRNA is going to depart, leaving behind its amino acid to incorporate into the protein. This marks the addition of one amino acid before proceeding to interpret our next codon. You might be wondering, how do we determine that a tRNA matching the UAC codon is going to carry isoleucine? Well, there's actually a codon chart that's going to become invaluable when you're trying to figure this out. It's not necessary knowledge for you to know for the ATI TEAS, but it is important for you to understand the mechanism and the keys that are taking place when we're talking about protein synthesis. Revisiting our mRNA sequence, let's decode the next codon, which is GAU. According to a codon chart, the sequence specifies that the next amino acid should be aspartic acid. The tRNA with its complementary anticodon, which would have been CUA, is going to bind to the codon. This tRNA is going to deliver that aspartic acid, and after the transfer, it's going to detach and connect to our original amino acid, forming our polypeptide chain. Typically, at some point, the mRNA sequence is going to conclude with a stop codon. So stop codons don't correspond with any amino acid. Instead, they signal the ribosome to tell them that protein synthesis process is done and complete, and they can release their amino acid. The outcome of the translation process is the creation of that amino acid chain. It's going to be assembled in a specific sequence that was dictated by our mRNA's code. However, it's crucial to remember that the sequence of mRNA is complementary to that of the DNA, making DNA the ultimate guide when it comes to protein synthesis. Despite DNA playing a pivotal role, the process cannot proceed without the crucial contribution of our mRNA, rRNA, and tRNA. After that amino acid is assembled, the protein may undergo further folding and modifications. It might need to be transported to a different location inside of the cell. These subsequent steps, depending on the protein's specific structure and function, will take place. So, practice question. Which component is essential for initiating the transcription of a gene into mRNA in eukaryotic cells? Is it A, DNA ligase, B, RNA polymerase, C, helicase, or D, ribosome? And the correct answer is B, RNA polymerase. RNA polymerase is crucial when it comes to the transcription process in eukaryotic cells, which reads the DNA sequence of a gene and synthesizes a complementary mRNA strand. DNA ligase is more involved in DNA replication and repair, not necessarily transcription. Helicase unwinds our DNA helix during replication, and ribosomes are the site of protein synthesis during translation, not involved in the transcription process initiation. So what is the role of mRNA in protein synthesis? Is it A, it brings amino acids to the ribosome? Is it B, it replicates DNA segments before protein synthesis? Is it C, it serves as a template for assembling amino acids into proteins? Or D, it transports proteins to different parts of the cell? And the correct answer is C, it serves as a template for assembling amino acids into proteins. So during protein synthesis, mRNA carries that genetic code from DNA during the transcription process to the ribosome. Here is where it's going to serve as a template for the sequence of amino acids in a protein during translation. tRNA, not mRNA, is responsible for bringing amino acids to the ribosome, so option A would be wrong. And then DNA replication in option B is a separate process from protein synthesis. So again, that doesn't make sense. And then lastly, with option D, transporting proteins to different parts of the cell is a function of other cellular components like our endoplasmic reticulum and our Golgi apparatus, not mRNA. For my entire life growing up, I've always been intrigued by the idea of owning a Sphinx cat. While they're available at select breeders, they're actually quite rare. Their survival in the wild ultimately seems unlikely. Consider picturing a Sphinx cat shivering out in the cold. It's a true testament about their specialized care needs. However, the genetics behind these hairless cats are utterly captivating, just like the cats themselves. Unlike their furry counterparts, Sphinx cats lack hair, a trait determined by their genetic makeup. Genes, which are segments of DNA, are passed down from the Sphinx kittens from both their mother and their father, dictating everything from their hairlessness to their distinctive appearance. An allele is a variant of a gene, often recognized by letters. For our discussion, we're going to use the letter F to represent fur. A Sphinx cat, known for its lack of fur, is going to possess two recessive alleles for the fur trait. Recessive alleles are typically indicated by the lowercase letter. Being recessive implies that this trait will only manifest if there are no dominant alleles present. Dominant alleles marked by their uppercase letter are traits that are expressed. In essence, they dominate over the others. Therefore, the Sphinx cat lacks the dominant fur gene, allowing the recessive trait of being furless to be expressed. So if we take a look at our guide here, we can see that a Sphinx cat is going to have two recessive alleles. Conversely, a long-haired cat genotype can either be two dominant alleles, meaning we have two uppercase letter Fs, or they can have one dominant allele and one recessive allele, meaning they'll have one uppercase F and one lowercase f. Because of that presence of just one dominant allele, that uppercase F, it's sufficient for that furry trait to be expressed, effectively masking that recessive allele. As we dive deeper into genetics, we come across a few terminology. Genotypes capital F, or lowercase f, are labeled homozygous genotypes, the prefix homo suggesting sameness, due to both alleles either being uppercase or lowercase respectively. If we break this down even further, our uppercase F is termed homozygous dominant, reflecting that uppercase notation, while our lowercase f is homozygous recessive, given its lowercase notation. A genotype with an uppercase F and a lowercase f is called heterozygous, where hetero in the beginning of that word means different, indicating that alleles are of different notations. One is uppercase and one is lowercase. If you were to observe a cat with hair, its genotype could either be homozygous dominant or it could be heterozygous. It's indiscernible just by their appearance. However, if we wanted to, we could test to clarify what the exact genotype is. In contrast, the genotype of a hairless Sphinx cat is always going to be homozygous recessive, since the presence of any dominant allele, which would be our uppercase F, would ultimately result in a furry phenotype. Let's tackle a monohybrid cross, where mono signifies the focus of a single trait, in this instance, fur. To analyze this, we are going to construct what we know as a Punnett square. It's divided into 4 sections, and in this instance, we're going to look at 2 heterozygous cats being crossed together. Our initial step, step 1, is we want to identify the parents' genotypes. In this case, we have 2 heterozygous cats. For step 2, we want to position one parent's genotype at the top of our Punnett square and the other parent's genotype on the side of our Punnett square. Then for step 3, we're going to proceed by crossing them, ensuring that whenever we have a dominant allele, that would be our capital F, we're going to list that first for consistency. So this is how we are going to cross them. We are going to multiply the top of the square by the side of the square. So our first example, we have F multiplied by capital F. So we would have a capital F, capital F. For our next, again, we're going to multiply the top of this square by the side of this square. And that is going to give us capital F, lowercase f. Then we're going to multiply the top of this square by the side of this square. And that is going to give us capital F, lowercase f. And then lastly, we're going to multiply the top of this square by the side of this square. And that is going to give us lowercase f, lowercase f. The resulting squares are going to reveal the potential genotypes of these cats' offspring. If you were asked on your TEAS what the offspring's genotypes would be, you could say that you have one cat that is going to be homozygous dominant, you're going to have 2 cats that are going to be heterozygous, and you're going to have 1 cat that is going to be homozygous recessive. This outcome can also be expressed as a 1:2:1 ratio or 25% capital F, 50% capital F, lowercase f, and 25% lowercase f, lowercase f. Now we're going to shift our focus to phenotype. But what exactly is a phenotype? The term phenotype can be remembered by associating pheno with physical appearance. So P in pheno, P in physical, that's how I like to remember that this is an observable characteristic of an organism. In this scenario, the trait in question is the presence or absence of fur. Given that the presence of at least one dominant allele, that would be our capital F, results in fur, offspring with the genotypes capital F, or capital F lowercase f are going to indeed have fur. Thus, 3 of our offspring are going to display that furry phenotype. Our homozygous recessive kitten, which is our lowercase f, ultimately lacks that dominant allele. So this cat is going to result in a hairless phenotype. Therefore, we can describe the phenotype distribution as 3 furry to 1 fur less, or we can even break it down into percentages. 75% are going to have fur and 25% are going to be hairless. It's crucial to remember that Punnett squares offer predictions based on probabilities, not certainties. They're ultimately going to indicate potential outcomes rather than guaranteeing specific results. For instance, while there's theoretically a 50 to 50% chance of a child being born male or female, many of us are familiar with families that have only daughters or only sons. These outcomes show the nature of probabilities as they forecast the possible events, adding an element of unpredictability. Until now, we've concentrated on only one single allele pairs, which is why we have been talking about monohybrid crosses, mono standing for one. However, cats possess a multitude of traits beyond just their fur status, each influenced by their own alleles. When we examine a cross involving 2 different pairs of alleles, we often refer to this as dihybrid cross. The prefix di indicates 2, so we're looking at 2 distinct traits. So I want you to picture this. There's a cat named Oreo, named for its black and white patched appearance, reminiscent of a cookie. Or admittedly, we just couldn't come up with a more original name. Now imagine beginning your day, walking into the living room to relax with your morning cup of coffee, only to find that your cat Oreo meticulously knocked your favorite mug off the coffee table. As you attempt to tidy up, you notice that Oreo is now pushing your pen off your desk. It's clear that not all cats are going to share in this behavior of playful disruption, but Oreo is irresistibly drawn to it. Day or night, he's irresistibly engaging in little gravity experiments. The origin of this behavior intrigues us. Is it genetic? Probably not. But let's practice dihybrid squares, imagining if playful disruption towards objects were genetic traits. Let's suppose that the enjoyment of causing playful disruptions is a dominant trait denoted by the allele capital D, and its absence, which is a more preference for calm, is a recessive trait symbolized by the allele lowercase d. Imagine that we have a cat that is heterozygous for both the fur trait and the playful disruption trait. Being heterozygous for both traits would leave them with a genotype of capital F, lowercase f, capital D, lowercase d. We're going to cross this cat with another cat that is both hairless and prefers calm environments. This means that they're going to have homozygous recessive traits. So they're going to have a lowercase f for the fur trait, and they're going to have a lowercase d for the playful disruption trait. Now, in Mendel's law of independent assortment, it suggests that traits are inherited independently of one another, indicating there's no genetic link between having fur and enjoying playful disruptions, for example. This means that a cat may enjoy playful disruptions regardless if they have fur or they don't. We're going to apply this principle to dihybrid cross. Here we have a setup of a 16-square Punnett square. Our very first step is we want to begin by establishing the parents' genotypes for our cross, which we have in our example here on the left-hand side of our screen, like we discussed before. Step number 2 is we want to determine the gamete combinations from each parent and place them alongside the top and the side of our Punnett square. But ultimately, how do we determine what letters to place there? We employ the FOIL method, which helps us figure out the possible gamete combinations. The FOIL acronym stands for first, outside, inside, and last. Let's take a look at each of these of how we determine our gamete combinations. Starting with our first cat, we are going to multiply the first letter of each one of our traits. This would be our capital F and our capital D, which would give us capital F, capital D. Next, we are going to multiply the outside of each one of our traits. That would be our capital F and our lowercase d, ultimately giving us this gamete formation. Then we have the inside. So we're going to multiply the inside of our formation here, which would give us lowercase f, capital D. And then lastly, we would multiply the last traits, which would be our lowercase f, lowercase d, ultimately giving us our final gamete combination. If you take a look at our last example, because everything is homozygous recessive, we don't have to do a whole lot of groundwork here because our gamete combinations are always going to be lowercase f, lowercase d. Now we're going to place these alongside our Punnett square. The uniformity of these combinations are going to highlight the limited allele variation that the parents contribute. And for step 3, we're going to merge the gametes to forecast the potential genetic outcomes that we're going to see with these cats' offspring. Just like we did with monohybrid cross, we want to maintain consistency when it comes to formatting. So if we have a capital F or a capital D first, we always want to make sure that we list those before our lowercase letters. We also want to make sure that we list our F's before our D's, because when we're looking at our combination of our gametes, that is how it's listed with each one of these cats. Ultimately, this method is going to ensure clarity and uniformity by interpreting the Punnett squares results. So just to give you an example, for our first square, we have capital F, capital D here, and we have lowercase f, lowercase d here. We are going to cross these two traits together. So we always want to start with F. So we have a capital F and a lowercase f. We want to make sure that capital F comes before the lowercase. And the same thing with our D. We have a capital D and a lowercase d. So we want to list the uppercase letter first and the lowercase letter last. So one more example we have here, same thing. We're going to cross the top of this square with the side of this square. So we have a capital F, lowercase f. We list that in order. And then look here, we have two recessive alleles. We have two lowercase d's. So we're going to list lowercase d and lowercase d. You're going to do this the entire time throughout the entirety of this dihybrid cross Punnett square. So we always want to know our genotypes and our phenotypes. So we're going to start with our genotypes. What genotypes are we going to have with these offspring? So if we take a look at our example, the distribution is as follows. We're going to have 4 out of 16 when it comes to capital F, lowercase f, capital D, lowercase d. We are going to have 4 out of 16 when it comes to capital F, lowercase f, lowercase d, lowercase d. We're also going to have 4 out of 16 of lowercase f, capital D, lowercase d, and again 4 out of 16 when it comes to lowercase f, and lowercase d, lowercase d. So the potential genotypes that we could see with these cats is a 1 to 1 to 1 to 1 ratio. Now let's shift our focus to phenotypes. So in this scenario, we see that about half of our cats are going to be furry and the other half of our cats are going to be furless. However, that's not the only thing that we're going to be testing with dihybrid cross. We also wanted to see the likelihood that the kitten was going to be born with characteristics similar to Oreo or not. So listed here with the use of polka dots, we can see that our pink polka dots, which is in this column, and this column show that there's going to be an 8 in 16 chance that they're going to be like Oreo. And with our blue polka dots in this column and this column, there's an 8 in 16 chance that they're going to be calm. If we break this down by their two traits, we can see that 25% are going to be furry and playful, 25% is going to be furry and calm, 25% are going to be furless and playful, and another 25% are going to be furless and calm. This ultimately leaves us with another ratio of 1 to 1 to 1 to 1 when it comes to our cat's phenotypes. It's crucial to note that in this particular case, the genotype and the phenotype ratios align, but this is not always the case. Whenever we're mixing two heterozygous cats together, we usually see a ratio of 9:3 to 3:1. But in this case, we had one heterozygous cat and one homozygous recessive cat. The key takeaway from this is that Punnett squares serve as a tool for estimating the probabilities of offspring inheriting specific genotypes or phenotypes. It's going to offer a glimpse of the intriguing dynamics of genetics. What links the color of flowers and human height together? They both serve as prime examples of non-Mendelian inheritance. This term refers to genetic traits that defy the conventional Mendelian principles where the presence of a dominant allele guarantees that expression of the dominant trait. Let's take a look at roses with the following three phenotypes: red, white, and an immediate hue of pink. This phenotype is known as incomplete dominance. In cases of incomplete dominance, there's a twist. The allele typically seen as dominant doesn't fully overshadow our so-called recessive allele when they coexist. In fact, the concept of dominant alleles becomes a little murky here. For instance, when the red flower genotype R, and the white flower genotype r result in offspring that are both R and r, they don't conform to that Mendelian expectation. Instead of that dominant R overpowering the recessive R to produce red flowers, it now actually manifests our pink flowers. This blending effect signifies incomplete dominance. Should two pink flowers be crossed, like we see here in our last example, this blending effect signifies incomplete dominance. Should we cross two pink flowers, like we see here in our last Punnett square, the offspring could be red, they could be pink, or they could even be white, showcasing the diverse outcomes when it comes to incomplete dominance. And lastly, we have codominance. So the prefix co in codominance suggests cooperation or working together, which aptly describe how the alleles interact in this scenario. Unlike with incomplete dominance, where one allele's expression is not entirely masked by the other, codominance involves both alleles being expressed equally and independently. To illustrate this, let's consider the genetic crossing of certain chickens. When a black chicken with the genotype B is crossed with a white chicken with the genotype W, the resulting offspring are going to exhibit the genotype B, W. These B, W chickens are going to display both black and white feathers with a speckled pattern. This simultaneous expression of both traits without blending or dilution amplifies codominance, a situation where both alleles contribute to the phenotype in a distinctly visible manner. So let's take a moment to consider our favorite food. While we all have our unique preferences, food fundamentally serves as a source of critical, large molecules necessary for life, known as biomolecules or macromolecules. These can include things like carbohydrates, lipids, proteins, and nucleic acids, each found in varying quantities across different foods. For instance, my favorite peanut butter is rich in both proteins as well as lipids. Although this video is going to reference examples of food containing biomolecules, our focus isn't going to be on nutrition, but understanding the major classes of those biomolecules and their critical roles in cellular structure and function. After all, these cells make up the organisms themselves. Before we deep dive into each one of these four different types of biomolecules, we must introduce an essential term, the monomer. The monomer acts as a building block. The smaller units composing any large molecule is termed monomers. As we explore each class of biomolecules, we'll discuss their respective monomers if they apply. Before we move on, let's take a moment to appreciate the stunning structure of biomolecules. As we review each one of these, let's think about how we might remember the primary and most common elements that they contain. There's a handy mnemonic called CHO, CHO, CHON, and CHUMP, where instead of an M, we actually use an N. This helps us actually recall the elements in each one of these biomolecules. Carbon is our C, hydrogen is our H, oxygen is our O, nitrogen is our N, and phosphorus is our P. So you may question, does this imply these are the only elements that we're going to find in these biomolecules? Certainly not. However, understanding the common elemental composition and their arrangements within these biomolecules is crucial because their structural configurations significantly influence their functions. Let's begin by exploring the class of biomolecules known as carbohydrates. Common sources like bread, pasta, fruits, and vegetables are all typically very rich in carbohydrates. The basic building block or monomer of a carbohydrate is known as a monosaccharide. A prime example of a monosaccharide is glucose. Glucose plays a critical role when it comes to cellular respiration, where it produces ATP, which is often referred to as the cell's energy currency. When two monosaccharides link together, they form what is known as a disaccharide. Here's a memory trick for you. You'll notice many sugars share a common suffix, OSE, like glucose, maltose, lactose, sucrose, and fructose, which helps indicate that these are carbohydrates. When many monosaccharides are linked together, they create a polysaccharide, which represents a more complex form of carbohydrate. If you're having any difficulty remembering which saccharide is which, I use the memory trick mono, di, and poly, because mono stands for one, di stands for two, and poly stands for many. Carbohydrates plays a crucial role in the structure and function of cells across various organisms. For instance, plant cells have cell walls composed of specific carbohydrates called cellulose. Similarly, fungi have cell walls made of a carbohydrate known as chitin, which interestingly is also the material that forms the exoskeletons of insects. A vital function of carbohydrates, such as glucose, is its role in energy production. Like we talked about before, glucose is instrumental in generating ATP, the primary energy carrier in cells, making carbohydrates a rapid and accessible energy source. Furthermore, this energy can be stored in the form of polysaccharides, complex carbohydrates like starch in plants and glycogen in animals, which serve as an energy reserve to be utilized when needed. Next, let's discuss lipids, which encompass fats and oils. Common sources include items like butter and olive oil, and they represent a remarkably diverse category of biomolecules, encompassing several types, including triglycerides, phospholipids, and steroids, just to name a few. While many lipids are composed of glycerol and fatty acids, this isn't universally true for all lipids. Lipids also do not have a true monomer because they do not have repeating units like we saw with carbohydrates. However, a common characteristic when it comes to most lipids is their hydrophobic nature, meaning that they tend to repel water. This hydrophobic nature means that lipids generally do not dissolve in water, unlike most other biomolecules, which makes them unique in their interaction with water and their role in the biological systems. Lipids play a crucial role in their structure and function of organisms and their cells. The cell membrane is the fundamental structures in all cells, whether it's plants, animals, or other organisms. This essential barrier is largely composed of phospholipids, arranged in a phospholipid bilayer thanks to their structural properties. Beyond structural roles, lipids are vital in long-term energy storage. While carbohydrates do provide quick energy, lipids store energy for longer periods, which organisms can tap into when carbohydrate sources are depleted. Lipids also play a significant role in insulation. This includes electrical insulation, such as the myelin sheath surrounding many neurons, which enhances the speed of electrical impulse transmission. Thermally, lipids are integral for temperature regulation, evident in the thick layers of blubber like we see in marine animals such as the harp seal, or even in an oversized kitty cat, which helps them retain heat in frigid temperatures. Another significant function of lipids is hormonal activity. Many lipids act as hormones, serving as key chemical messengers that regulate various biological processes throughout the body. Next up, we have proteins. Common protein sources include meat, nuts, beans, and even eggs. Proteins are polymers made up of amino acids, which serve as their monomers. There are various types of amino acids which link together to form proteins. Proteins are crucial for both structure and function of an organism and their cells. Structurally, proteins are major components of tissues such as muscles. They are also found in other body structures such as hair and collagen. Proteins play an essential role in cell membrane integrity by forming protein channels and receptors, which are vital for transporting substances across the cell membrane and for cell signaling, enabling these cells to communicate and coordinate their actions. Functionally, most enzymes are proteins. Enzymes catalyze biochemical reactions in the body, aiding in both the synthesis and breakdown of substances during metabolic processes. Proteins are also fundamental to the immune system. Antibodies produced by immune cells are proteins that play a key role in defending the body against pathogens. Furthermore, several critical hormones, including insulin, are also proteins, highlighting their role in regulating physiological processes. When it comes to genetics, although genes are composed of DNA, many genes encode for proteins, underlying the genetic basis for building and regulating the body's proteins. This brings us to our final category, nucleic acids, which encompass both DNA and RNA. We'll explore nucleic acids in greater detail when we talk about genetics. The monomer of a nucleic acid is known as a nucleotide, in terms that conveniently resemble nucleic acids, making it much easier to remember. Nucleotide, nucleic acids. Nucleic acids are essential across all life forms, whether it's plants, animals, fungi, or even bacteria, as they contain the genetic instructions necessary for introducing cellular activities. In terms of their importance inside of your cells, nucleic acids hold all your genetic information in the form of DNA and RNA. This genetic content is crucial for coding your traits that define your structure and your function, influencing everything from physical appearance to psychological processes. So our first practice question: what is the monomer of carbohydrates? Is it A, nucleotide, B, amino acids, C, fatty acid, or D, monosaccharide? And the correct answer is D, monosaccharide. Remember, monosaccharides are the simplest form of carbohydrates, and they serve as the monomers for those more complex carbohydrates. What type of lipid is primarily used by the body for long-term energy storage? Is it A, steroids, B, phospholipids, C, triglycerides, or D, waxes? And the correct answer is C, triglycerides. Remember, triglycerides are the main form of stored energy in your body and are found in a lot of your fat cells. Which type of macromolecule plays a key role in forming cellular membranes? Is it A, nucleic acids, B, carbohydrates, C, lipids, or D, proteins? And the correct answer is C, lipids. Lipids, specifically phospholipids, are fundamental components of cell membranes. Now we're going to discuss infectious agents, often referred to as microorganisms. However, it's crucial to note that not all agents that we're going to cover today qualify as living organisms. Some actually aren't even alive at all. They're neither cells nor considered life forms. So for clarity, we're going to refer to them as infectious agents instead of microorganisms. These agents can enter our bodies, which act as hosts and potentially cause injury, disease, or even death. When discussing anything that can cause disease or injury, we're going to use the term pathogenic. This term can be broken down into two parts: patho, which means disease, and genic, which means causes. Thus, pathogenic refers to an infectious agent capable of inducing disease. Another term that you might encounter when you're taking the TEAS is virulence, which describes the severity or harmful potential of a pathogen. It's important to understand that virulence doesn't necessarily correlate with more damaging outcomes. So for instance, a more virulent strand of a virus may not always cause more severe disease, but could be more adept to invading immune systems, more transmissible, and it could even be more capable of circumventing vaccines, or in the case of bacteria, antibiotics. So in summary, pathogenic means capable of causing disease, while virulent refers to the potential severity or nastiness of a pathogen. So let's start off with our favorites, viruses, familiar to many and very significant in the study of microbiology. Viruses are tiny entities ranging from anywhere between 20 to 400 nanometers in size. Notably, viruses are not cells and they're not actually considered alive. They remain motionless on surfaces like benchtops or the ground outside of a host organism. To be active, a virus has to enter a host, whether it's an animal or it's a plant, and once it's inside, it behaves as an intracellular obligate parasite. This means that it's compelled to reside within a host cell to function, specifically to replicate and perform activities. Viruses are classified based on structural components as well as genetic material. The outside shell of a virus is known as a capsid, and the genetic core inside is referred to as a genome. Thus, viruses can be categorized by their capsid structure and whether their genome consists of DNA or RNA. Additionally, some viruses have an envelope, which is derived from the host cell's membrane as the virus exits the cell, enveloping itself for protection and aiding in infection. The impact of a cell on the body and its pathogenicity depends on where it infects. Different viruses target specific regions, such as the respiratory tract, your gastrointestinal tract, systemic, skin, central nervous system, just to name a few. For instance, the common cold, influenza, and COVID affect the respiratory system, while rotavirus and mumps is prevalent in your GI tract, especially in children. Systemic infections like chickenpox and HIV can affect the entire body, and skin-specific viruses like we see with HPV can cause cervical warts, or even worse, cervical cancer, due to that cellular mutation induced by viral genetic material integration. Viruses attach to a host cell via structures like spike proteins on their capsid, which bind to specific receptors on the cell membrane. Upon attachment, the virus injects its genetic material into the host cell, hijacking the cell's machinery to replicate new viruses. This is often going to lead to the host cell's death, contributing to the disease. In some cases, like we see with HPV, that integration and mutation of viral DNA can lead to cancer. Fortunately, though, for many of these viruses, vaccines are available, providing protection by priming the immune system against these infectious agents. Moving on, let's talk about bacteria, which marks a significant jump in scale. Bacteria can range anywhere from 0.2 to 15 microns in size. To put this into perspective, 0.2 microns is equivalent to 200 nanometers, and 15 microns is equivalent to 15,000 nanometers. From here until the rest of our examples of microorganisms, unlike viruses, we're dealing with living organisms, cells that are very much alive. Bacteria are classified as prokaryote, meaning that they have DNA, but it's not contained within a membrane-bound nucleus. Instead, it resides in an area called a nucleoid. Unlike eukaryotic cells, most bacterial organelles are not enclosed by membranes. A key structural feature of bacteria is their cell membrane. It's similar to that of other cells, but more permeable, which means that they are more susceptible to swelling and bursting under fluid pressure. Fortunately, bacteria have a protective cell wall, which helps maintain its structural integrity against such pressures. This cell wall is crucial for the bacteria's survival and is composed of a network of proteins and sugars. Bacteria can be further categorized based on the composition of these cell walls into two types, gram-positive and gram-negative. Our gram-positive bacteria have a thick cell wall, while gram-negative bacteria has a thinner cell wall but an additional outer membrane. Additionally, bacteria can be categorized by their shape. Cocci is more spherical and bacilli is more of a rod shape. The organization of these shapes can be indicative. For example, cocci that group in a grape-like structure can be known as staphylococci, and those that form in long chains are known as streptococci. One disease example could be chlamydia, which is a bacteria that requires intracellular environments to access energy and carry out its functions effectively. As a result, they can also lead to cellular damage, ultimately causing scarring. Such scarring can occur in the reproductive tract, especially in females, potentially leading to infertility. In the eyes, this scarring can actually impair vision and lead to various types of visual dysfunctions. Another example would be mycoplasma, notably one of the primary causes of community-acquired pneumonia. Mycoplasma is unique because it lacks a typical bacterial cell wall, which allows it to live both inside and outside of host cells. We also have bacteria like Streptococcus, which can cause conditions such as strep throat, primarily affecting the respiratory tract. Staphylococcus aureus, aureus meaning golden, which is a common cause of skin infections. Additionally, there is Clostridium tetani, which is particularly interesting because it produces exotoxins. Gram-positive bacteria like Clostridium tetani release exotoxins that can cause severe damage by infecting motor neurons with skin wounds, leading to tetanus. This condition is life-threatening and it can cause muscles to stiffen uncontrollably. In contrast, gram-negative bacteria produce endotoxins, which are part of their outer membrane. When these bacteria die and break down, they release those endotoxins, triggering intense inflammation as well as other serious health issues to the host. As we progress to larger cellular structures, we encounter fungi, which range in size from 2 to 200 microns. Fungi, like the cells we find within our body, are eukaryotes, meaning they possess membrane-bound organelles, including nuclei. Unlike bacteria, fungi have cell walls made up of chitin, which is a polysaccharide also found in the exoskeletons of insects like spiders. Fungi has the capability to reproduce both sexually and asexually, often relying on spores, which function similarly to seeds to spread and propagate. When fungi causes diseases, the condition is referred to as mycosis, which can manifest as superficial or deep infections. Superficial fungal infections primarily affect the skin, typically arising from fungi that releases enzymes irritating the skin and causing symptoms like itching and redness. A common example is tinea, which is known as ringworm, thought to not be actually a worm but a fungal infection. Depending on the affected area, tinea may be called tinea capitis if it's on the scalp, or tinea pedis if it's on the foot like we see with athlete's foot. Fungal infections can also impact mucous membranes, leading to a condition known as thrush, which can occur in our mouth. We have oral thrush, or it can occur into the genital area known as vaginal thrush. More severe and deep fungal infections may involve internal organs, like the lungs or bloodstream, particularly in immunocompromised individuals. In such cases, fungal infections can lead to serious health complications and are a common cause of mortality in people with weakened immune systems, such as those with HIV. Protozoa, also referred to as protists, range in size from 1 to 50 microns. These organisms are eukaryotic and usually unicellular like we see with fungi, or they're typically multicellular. Protozoa possess all the organelles necessary for metabolic functions, similar to those found in our cells, including nuclei and mitochondria. Examples of diseases caused by protozoa include malaria and giardiasis. Malaria begins with the protozoan's life cycle inside the mosquito's gut and saliva. When a mosquito bites, it injects what can be similar to cysts into the bloodstream, which then travels down into our liver. Here, they mature within the hepatocytes before bursting out and infecting red blood cells. Within these cells, the protozoa reproduces asexually, leading to the typical symptoms we see with malaria. Once released from the red blood cells, the protozoa enter a gamete form, ready to be picked up by another mosquito, continuing that transmission cycle. It's important to note that the mosquito only serves as a vector. The actual infectious agent is the protozoa. Giardiasis, on the other hand, is a digestive disease which enters the body through contaminated food and water. The cysts are ingested and travel to the upper digestive tract where they hatch and multiply through mitosis. They then attach to the digestive tract walls, absorbing nutrients and causing irritation and inflammation. This process often results in symptoms like severe, watery, and sometimes bloody diarrhea. Eventually, the protozoa produces more cysts, which are excreted in feces and often contaminate food and water sources, perpetuating the cycle of infection. Finally, we move on to animals that can be classified as helminths, which are worms or ectoparasites. Helminths are multicellular eukaryotic organisms that range significantly in size from as small as 3 milliliters all the way up to as large as 10 meters. In terms of classification, helminths can be divided based on their shape. They can be roundworms, flatworms, or flukes. Roundworms, such as hookworms, typically inhabit the gastrointestinal tract and have a cross-section circular appearance. Flatworms, like tapeworms, are often very long and have a ribbon-like shape. Flukes, such as liver flukes, are typically leaf-shaped. As for ectoparasites, these include organisms like mites, fleas, and bedbugs. These parasites are primarily known for causing skin irritation and inflammation. They attach to the skin, feeding off the host, and often leading to discomfort and allergic reactions. If we consider creatures like ticks, they can transmit bacteria through their bites, known as Lyme disease. A tick bite introducing bacteria into the skin can potentially lead to significant health issues. Which of the following is characteristic of fungi? Is it A, they are photosynthetic, B, they reproduce by binary fission, C, they have a cell wall made of chitin, or D, they are typically motile? And the correct answer is C, they have a cell wall made of chitin. Remember, fungi have that cell wall, and it's going to distinguish them from plants who have cell walls made of cellulose. Which type of microorganism causes malaria? Is it virus, bacteria, protozoa, or fungi? And the correct answer is C, protozoa. Malaria is caused by protozoa, which is transmitted to humans from a bite of an infected female mosquito. What is a unique feature of viruses compared to other microorganisms? Is it A, they contain both DNA and RNA, B, they require a host cell to replicate, C, they have a rigid cell wall, or D, they can undergo photosynthesis? The correct answer is B, they require a host cell to replicate. Remember, viruses are unique in that they cannot replicate on their own. They have to invade a host cell and use its machinery to produce new virus particles. Infectious and non-infectious diseases represent two broad categories of illnesses, each with distinct causes and mechanisms. Infectious diseases are caused by pathogenic microorganisms, such as bacteria, viruses, parasites, or fungi. And these diseases can spread directly or indirectly from one person to another. For example, COVID-19 primarily spreads through respiratory droplets when an infected person either coughs, sneezes, or talks. It can also spread by touching surfaces contaminated with the virus and then touching your face, although it is less common. Similarly, influenza is caused by various strands of the flu viruses and is highly contagious, spreading through respiratory droplets. Chickenpox is another highly contagious disease, most commonly affecting children. This virus spreads through direct contact with a rash or through the air by respiratory droplets when that infected person coughs or sneezes. Non-infectious diseases, also known as non-communicable diseases, are not caused by infectious agents and cannot be transmitted between people or other organisms. These diseases are often the result of genetic, environmental, or lifestyle factors. Examples can include diabetes, which results from the body's inability to produce or effectively use insulin. Heart disease is often caused by a combination of dietary, genetic, and lifestyle factors leading to the buildup of plaque in our coronary arteries. Cancer can have various causes as well, including genetic predisposition, environmental exposures, and lifestyle choices such as smoking and diet. understanding the distinctions between these two types of diseases is crucial for effective prevention, treatment, and management strategies. Next, we're going to examine the various ways diseases are transmitted and often referred to as modes of transmission. The first category we are going to discuss is going to be direct transmission. This occurs when infectious agents are immediately transferred from a source or reservoir, such as an infectious person, directly into a susceptible new host. Direct transmission can occur in two primary ways, either through direct contact or droplet spread. Direct contact is going to involve physical interaction with bodily fluids from an infected individual, which might include oral secretion, blood, or other fluids like we see with skin lesions, semen, or vaginal secretions. This type of transmission can result from activities like touching, biting, kissing, or sexual intercourse. Droplet spread, another form of direct transmission, requires droplets from respiratory droplets containing those infectious agents. This typically happens when an infected person coughs or sneezes, projecting droplets directly onto someone nearby. Diseases like pertussis, also known as the whooping cough, is often spread this way. For droplet transmission to occur, close proximity is necessary, generally less than 2 meters, such as sharing a small space like a car or a room. It's important to understand that both direct contact and droplet spread are considered direct transmission methods. Next, we'll discuss indirect transmission, which includes several mechanisms, one of which is airborne transmission. Airborne transmission involves infectious agents that are carried on dust particles or within droplet nuclei that remain suspended in the air. Due to their small size and light weight, these droplet nuclei can linger in the air for extended periods of time, allowing them to be inhaled into a host's respiratory system. With airborne transmission, neither direct contact nor close proximity is necessary for the disease to spread. The droplet nuclei can travel and disperse widely across an environment and can infect a host without any direct interaction with the person who was infected. Indirect transmission involves objects and items, often referred to as fomite transmissions. They occur when infectious agents are transferred from an infected person to an object or a surface. These objects, like door handles, tabletops, utensils, or electrical devices, can then harbor the infectious agents for varying lengths of time. When another individual touches these contaminated surfaces and then touches their face, eyes, nose, or mouth, they can inadvertently introduce that pathogen to their body, leading to an infection. The risk of fomite transmissions depends on several factors, including the type of pathogen, the environment, and the material of the object. For example, hard surfaces like plastic, stainless steel can retain some viruses for days, whereas more porous materials like fabric are less conducive to long-term survival of viruses. Regular cleaning and disinfection of frequently touched surfaces are effective strategies to reduce the risk of disease spread through fomites. Hand hygiene is also crucial in preventing the acquisition and spread of infections through indirect contact with contaminated objects. And lastly, we have vector-borne diseases. They are transmitted when an infected person passes a pathogen to a vector, typically an insect, which then spreads the infection to another person. Without this vector, the disease cannot pass directly between individuals. Due to their complex transmission dynamics, these diseases are among the most challenging to prevent and control. For example, preventive strategies in the fight against malaria, which is spread by mosquitoes, includes vaccinations and environmental tactics. These environmental methods often involve using mosquito nets or removing standing water to disrupt the mosquitoes' breeding grounds. Finally, let's talk about microscopes. To start, it's essential to grasp a few key terms when understanding how microscopes work. Magnification is the primary concept often associated with microscopes, highlighting their ability to enlarge small objects into observable sizes. Another critical term you're going to need to know is resolution, and that's the ability of a microscope to distinguish between two closely positioned objects as separate entities. Without adequate resolution, two proximate objects might appear as a single blurred entity. For instance, a microscope with a resolution of 0.2 microns requires objects to be at least 0.2 microns apart in order for them to be distinct from one another. Let's explore various types of microscopes, each designed with both magnification and resolution in mind to effectively visualize and study specimens. First up, we have the light microscope, which is the type of microscope you typically see in biology classroom settings. As the name suggests, these microscopes utilize light to visualize specimens. The most common type of light microscope is the bright field light microscope, which typically displays a darker image against a light background. Alternatively, there are dark field light microscopes, which includes a component known as a light stop. This element blocks most of the direct light that we see through the light source. And because of this, the only light that reaches your eyes is the light that has been reflected or refracted by structures within the specimen themselves. This technique creates a visual effect where lighter images appear against a dark background, enhancing the contrast and detail of the specimen. Light microscopes utilize light to visualize specimens, but not all microscopes are going to operate by this principle. For instance, to observe extremely small entities like viruses, which are typically smaller than your average bacterium, more advanced technology is necessary to enhance magnification and resolution. This is where electron microscopes are going to come into play. Electron microscopes use beams of electrons instead of light to create images of the specimens. Among electron microscopes, we have two. We have transmission electron microscopes, known as TEM, which are particularly effective in examining the internal structures of a specimen. And on the other hand, we have scanning electron microscopes, known as SEM, which are better suited for detailed visualization of three-dimensional structures of a specimen. Let's finish off with some practice questions. Which microscope would be best for viewing the detailed external morphology of a microorganism? Is it A, light microscope, B, transmission electron microscope, C, scanning electron microscope, or D, fluorescence microscope? And the correct answer is C, scanning electron microscope. Remember, SEMs are ideal for examining surface structures in high detail as they scan the surface with a focused beam of electrons, providing us with a three-dimensional image. What type of transmission involves infectious particles suspended in the air over time and distance? Is it A, direct contact, B, droplet transmission, C, airborne transmission, or D, fomite transmission? And the correct answer is C, airborne transmission. Remember, our airborne transmission refers to the spread of infectious agents through droplet nuclei that are going to remain infectious and suspended in the air over a distance and time beyond 1 meter. Which disease is known to be transmitted through droplet spread? Is it A, HIV, B, hepatitis C, measles, or D, influenza? And the correct answer is D, influenza. Influenza is well known to be transmitted through droplets that are expelled from an infected person, either coughing or sneezing, requiring close proximity to spread. And that's everything you're going to need to know. Biology, right? As always, if you have any questions, make sure that you leave them down below. I love answering your questions. Head over to NurseCheungStore.com, where there is a ton of additional resources available to you to help you ace those ATI TEAS exams. And as always, I'm going to catch you in the next video. Bye!